Assalamualaikum and hi. Welcome to my Pita 2 presentation video with the title The Effect of Enriched COPEPODS on the Coloration and Feeding Rate of Beta-Splendens. My name is Siti Nadhirah binti Muhammad Nasir with MATRIC number S48372. I am the supervisee of Dr. NURUL NADIAH binti RASDI. For my experiment, it is between the correlation of COPEPODS and Beta-Splendens. Copepod is a tiny zooplankton with a range size of 0.5 to 5.0 mm. It contains high level of fatty acid and have natural carotenoid pigmentation. Thus, it can help in enhance the color in ornamental fish. Copepod also act as a major component of marine zooplankton in terms of biomass, diversity, and abundance. For beta-splendens, it is one of the most popular and have high commercial value in aquaculture ornamental fish in the global trade. They are also fancy with unique body shape and color combination. The objective of my experiment is to study the effects of different COPEPODS enriched diet on the coloration and feeding rate of beta-splendens. Problem arises when the commercial food do not fit the size and nutrition level needed by the fish. High demand of betta fish in ornamental fish industries led to increasing number of demand in live feed. Enhancing the color of fish by using hormones and additives affects the water quality and harms the fish life itself. The color enhancement in a wide variety of fish species have been achieved with the diet supplementation with the pure or extracted pigments from live feed. Copepod mainly feed on yeast because it have low in polyunsaturated fatty acid PUFA contents. Live food organisms contain all the nutrients such as essential proteins, lipid, carbohydrates, vitamins, minerals, amino acids, and also fatty acids. Moving on to the materials and method, the first thing first is the copepod collection and culture. All of the process was done in the hatcheries and the water quality parameter was being measured every 2 days. For beta-splendens breeding and rearing, each treatment contained triplicates and there were 10 fries in each treatment. Also, the water quality will be measured every 2 days. For the copepod enrichment procedure, there will be 5 different enrichment ingredients such as spinach and carrot, microalgae chlorella species, rice bran, capsicum, and yeast. All of them will be enriched to the copepod in 24 hours time before feeding to beta-splendens. For feed intake, it was done by examining how many copepods that can be consumed by the beta-splendens in the meantime. For the coloration of beta-splendens at the end of 60 days feeding treatment, the color intensity of the beta-splendens will be measured by using conical minolta chrometer in a measurement of International Commission Illumination CIE. For the proximate analysis of copepods, lipid and protein content of enriched copepod was done. For proximate analysis result, copepod enriched with capsicum hold the highest protein content, which is 61.94. Meanwhile, copepod enriched with chlorella species microalgae hold the highest lipid content, which is 3.78. For the coloration result in L value, we can see that from the table it is dark when L value equals to 0 and white value when L equals to 100. Rice bran hold the result nearest to 0, which is 30.98. Rice bran enriched copepod contain hormone that can enhance pigmentation called eumelanin. Skin pigmentation occur on the fish skin enhanced by a black pigment called eumelanin. Eumelanin hormone produces black coloration and dark brown color in fish skin during pigmentation. That is why beta-splendens fed with rice bran enriched copepod rise to the black color in the skin pigmentation. Moving on to the A value of the coloration. It is green when the A value is negative and it is red when the A value is positive. We can see that from the table below, the mixed vegetable carrot and spinach hold the highest value, which is 68.86, which is red. Fish fed with mixed vegetable carrot and spinach contain natural astaxanthin develop more of red color. Natural sources of beta-carotene that can be found in the carrot and spinach help in the color intensity and prominence of red-orange color increase in the fish. For the B value result, it is negative when the color is blue and is positive when the color is yellow. We can see that from the table, capsicum hold the highest positive value, which is 89.17. Fish unable to produce color of certain types of colors such as yellow. Capsicum contain natural available carotenoid-rich ingredients such as xanthophylls. Xanthophylls can enhance the skin color of fish to yellowish. Beta-spendent fat on rice bran enriched coffee pod reads the negative value, which is negative 7.21. Rice bran contain free retinol, making there is coloration shift in the fish body, giving the negative reading. Thus, the fish produce blue in color. For the feeding rate result, beta-spendent fat on mixed vegetable, carrot and spinach enriched coffee pod reads the highest feeding rate, which is 1.65. Mixed vegetable is organic basis food, means it is safe food for fish. High protein and lipid content in the enrichment maximize the feeding rate of beta-spendent because it can support the metabolic energy of the fish. Fish can utilize plant feedstock carbohydrate for energy. In conclusion, the effect of enriched coffee pods on the coloration and feeding rate of beta-spendent was a success as the enrichment types given to the beta-spendent affect the coloration and feeding rate of the juveniles. This study suggested that it can be practiced by the ornamental fish sellers to get high income in trading the fish. This is the Gantt chart of my experiment. References. That's all from me. Thank you.